So our last talk is from Dr. Joseph Larkin's lab, and we're talking about sample preparation. And in this case, we'll be talking about sample preparation of plants. So is Joe here? Oh, you're, oh, yeah. Well, so Joe will introduce his students.
Hi, everyone. I'm Joe Larkin. I'm an assistant professor of biology and physics. And just so I don't take up too much time, my student, Michael Zulch, is a PhD student in the molecular biology, cell biology, and biochemistry PhD program. And through the SB2 fellowship, he did a internship at Join, which is a local biotech company that's trying to engineer root microbe interactions. And so based on his experience doing that kind of work, he came up with a project that he thought was perfect for the DAMP Lab pilot program. I agreed, and Michael will tell you about it now.
Thank you very much for having me to speak today. I have the distinct pleasure of keeping you from lunch, so I'm going to try to keep it. I'm going to try to get you there as quickly as possible, but let's not jump over too much cool stuff. What you're looking at right now is probably what I would argue one of the most important interfaces for life. It is the interface of the root, which protrudes from the bottom right of this picture, and then the bacteria that surround the root, which are GFP. This is a confocal image. So yeah, indeed, the bacteria play a really important relationship with plants and just life in general. It's more often than not a mutualistic relationship in which the bacteria provide critical things to the plant, such as fixed nitrogen and nutrient uptake of, for example, phosphorus, and prevent diseases from inflicting the plant. And this is a mutualistic relationship, and the energy provided to the bacteria to do this comes from the sun and is photosynthesized into the ground through the plant. So this is kind of the area where a lot of soil ecologists and synthetic biologists have high interest, particularly those who want to try to engineer and improve this relationship. So the question, though, becomes realistically, how many bacteria can live on a plant? And this is one of the fundamental questions of my dissertation, is trying to resolve what is called the carrying capacity and the microbial carrying capacity of the plant. So to do this, you see this? No, I use these little tubes. Sorry, I use these little tubes to grow a single plant, and then after the plant grows to a certain point, there's carbon in the system, and I inoculate it with bacteria. And when at time 0, you see in the middle, when I inoculate it with bacteria, over time, the bacteria grow to a saturation point. And this saturation point can be thought of as the carrying capacity of the plant, basically how many bacteria can live on the plant. From an engineer's perspective, how many bacteria you have serving your function. But the problem with engineering, though, is that what it does is take energy or carbon that it would normally use to convert into biomass, and instead diverts that into the production of a protein, or in the case of the nitrogen fixing genes, a lot of the energy required to fix nitrogen is costly, and so it diverts some of the energy resources away from it. And so what I'm very interested in doing is trying to figure out how these energy engineering strategies actually affect the carrying capacity of the plant. And here, added to the previous graph that you saw, are just like two theoretical lines which demonstrate a 20% reduction in carrying capacity as the blue line, and a 80% reduction in carrying capacity being green. I want you to focus on the conservative estimate of the fitness cost of engineering strategies being 80%, because you can see that it overlaps with the standard deviation of the wild type bacteria. And this problem would be difficult to solve unless you had high replicates. Based on this data, a power analysis would suggest that I would need 50 to 80 replicates per group to resolve this carrying capacity difference. And this is really where the DAMP Lab comes into play, because while I can grow these plants in a relatively small profile, and grow a large number of them. The task of counting bacteria can get pretty multiplied pretty quickly. And for example, if I were to just measure 5 different strains of bacteria that express a protein at 5 different levels, it would amount to some 1,250 to 2,000 agar plates to get that power that I need to be able to demonstrate that there is a fitness effect in the between these two populations. And so my interest in working with the DAMP lab was really in trying to get away from that conventional method of counting bacteria on a plant and pivot to something a little bit more sophisticated like qPCR. And based off of what we knew about the DAMP lab, we knew that they were like the go-to people in terms of like expertise in nucleic acid extraction and qPCR automation, which we knew would help us avoid pitfalls of this project and implement best practices resulting in higher quality DNA. The standardization and repeatability was key too, because we will be operating at such a high level of sample processing. But another benefit of this too is that as new members join the lab, maybe they're undergrads, or we recruit a lot from physics too, who don't oftentimes have the lab skills to design the experiment, at the plant microbial interaction level, and then just pivot over to the molecular level so easily. We know can be a consistency between these users when it comes to like how this data is produced, which is a big plus for us. And then, as I was mentioning, an increased throughput of samples was also important because it can help speed up progress and explore more hypotheses and reduce physical strain selfishly is something that I was hoping for. But also, like Joe said, I did work at Join, which was a subsidy of Ginkgo Bioworks. And so I was like well in tune to the whole high throughput robotics way of doing science. And I knew as a trainee from that perspective, this could be valuable for my career development. So I was really excited to submit a pilot to the DAMP lab. And these are some of the components of what we submitted. We wanted to develop and implement an automated protocol for DNA efficient extraction and PCR testing of plant microbe samples. The deliverable was going to be a workflow for the seamless submission of the samples and the data return ultimately. We wanted in this pilot to validate that we could submit samples of what we knew the composition of was into the process and get that back through the data to work towards these benefits of reduced work burden, expert handling, and ultimately leading us to the ability to ask these more frequent, high quality questions. So the services that I utilized were sort of off the shelf, I guess you could say, in terms of they were really just DNA extraction and PCR. We knew that the PCR component based off of the COVID testing was probably going to be an easy thing. If I gave them DNA, we could confidently say they could amplify it. However, the DNA extraction was a little bit more of a concern for us because I do work with a more non-standard sample type here at BU. Plant material is notorious for having certain inhibitors of PCR that could prevent amplification of DNA, as was mentioned in the previous talk. And the isolation part actually, because the plants have tough cell walls, requires this like bead-based system to kind of massacrate the samples and disrupt the tissues. So together, Ty, Courtney, Dave, and I like looked at this process, and we kind of like this is the typical QIAGEN kit, which I think the microbiome, a lot of people in the microbiome initiative use too. It's a spin column based kit in which we reasoned that the only real two important components of this workflow were the lysis part and the contaminant removal part, mainly because those were the only parts of the kit that were specific to plant tissue. They were able to lyse the tough cell walls and also remove the tissue-specific contaminants. And so what we just kind of tried to do was take it from here. We realized after we removed the contaminants, what we had was DNA in an aqueous solution, and that the DAMP lab was already well equipped to remove that DNA from the aqueous solution using their magnetic bead-based DNA isolation method. And it worked quite well. It worked really well on the first pass. And here are some statistics on how the labor was reduced, though. We went from a process that took 4 hours and with 2.7 hours of manual time down to basically minutes of prep work and then run it on the robot. But I think a key element about this slide here is that whether or not it was reduced by 75% or 25%, effectively for me as a researcher, it was reduced 100% because I can really just submit the samples even today. And if they were working tomorrow, they'll be here. They could process them maybe tomorrow. So it is a real big benefit to have a service like this here at BU. And you can see there was a lot of development work with the Hamilton robots, likely by Dave. Okay, so like what were our goals? Well, our goals were obviously to get all these data points, right? So in our first pass, our first high volume pass of this run, we submitted 152 samples, and we only had 14 extractions fail, which I think is pretty good because we didn't do any troubleshooting to try to like improve that 9% extraction rate. Of the successful extractions, we did qPCR, and only one of our reactions failed out of range out of the entire sample set. And I think, you know, this isn't the most impressive data point ever, but I think it speaks to the system. The qPCR instruments issue a quality score for every data point. And when I did this extraction and qPCR completely manually, you can see that while only marginally smaller, the automated method performed significantly better, as you can see by the p-value here. So as part of this project, the 152 samples that we submitted, I created kind of like mock samples to try to make sure that what went into the protocol was what we got out in the data. And so I essentially I took a tube, put a leaf in it, and added 2 volumes of bacteria. I then diluted one of those bacteria populations by half 9 times, and in 8 replicates submitted it to the DAMP lab. And what you can see is what I expected here is on the x-axis, and the results that I got in terms of relative proportion of the 2 species correlate very nicely with one another. So to conclude, we've effectively developed this protocol. You know, it's ready to use the next time I have samples available to me. It significantly cuts processing time and labor time for me. It has improved quality observed in the reads, and on a first pass extraction rate failure of only 9.2%. And our validation experiment went really well. So now we have this tool available to us to ask and figure out how many microbes a plant can support. So thank you so much for allowing me to talk today.
Question. Oh, go ahead. So and this might be tied with that, but is the protocol that is developed now, is it specific to the types of plants you work on, or is it?
I mean, you definitely would have to test it, but the only, like I said here, like the QIAGEN kit is the first 2 steps that we use from that kit in this process, and it has to do with the reduction of inhibitors and the lysis of the tissue. And that kit is compatible with a large number of different plant samples, model organism and non-model organisms. And so we just use those 2 elements to create a solution that's ready to be put into your DNA isolation pipeline. And it worked really well, surprisingly well.